Hello everybody, my name is HiBB and we are at University of Massachusetts, Boston presenting this research, which the main goal of this research is to detecting chromosomal structural variation using Jaccard distance. We are using a parallel architecture in order to achieve this goal. At first I'm going to talk about the introduction, then we're going to jump into the proposed method. I'm going to go through each step of the pipeline. I'm going to introduce what is the binary fingerprints and how we implemented using SSE instruction and came up with a parallel method to compute the Jaccard distance. We're going to go through the dataset and discuss the results then and impact of our method, and we're going to end up with conclusion. So one of the main challenges for cancer therapy is that the gene mutation might be unique for each patient, and also several patients might have different responses, show different responses to the same therapy. So the personalized cancer therapy is the answer to this problem, and the keys to the personal cancer therapy is to find the DNA mutation. And finding the DNA mutations is so difficult because the challenge is that the large amount of data produced by the sequencing methods. We know that the DNA, the human DNA, has more than 3 billion pair bases, and the structural variations inside these pairs are a medium to large scale chromosomal rearrangement that can affect large area of the DNA. And they found in a disease like cancer, female fertility, and other diseases. In this picture, you can see different kind of SVs like deletion, inversion, insertion, and translocation. In order to find these SVs and overcome this challenge of searching in a large amount of the data, related methods try to solve this problem by different methods. For instance, Delhi, which introduced at 2012, integrate short insert paired ends and long range mate pairs and split read alignments to accurately find out the SV's location. Another method is TIDE, which introduced at 2014. This method used low quality sensitive hashing to reduce dimensionality, and this method is an approximation for finding the similarities between paired reads. Another method is Lumpy, which is introduced at 2014, is a novel SV discovery framework that naturally integrates multiple SV signals and join them across multiple samples. Another method is TARDIS, introduced at 2017. This method combines multiple read signatures into a single package to characterize most SV types simultaneously, and it is not appropriate for detecting interchromosomal translocation, which in this research we want to aim this goal. So the proposed method of this study used Jaccard distance to measure the similarity of two DNA sequences, and the reason we can use Jaccard distance is that the DNA sequences are a string of characters. So similar methods use approximation like LSH to find the similarity between the pairs. But in this research, with using the advances in multi-core parallelism, for instance SSE and ABX instructions, we can detect the SVs using exact Jaccard distance without restoring to approximation. So the two main goals of this research is to detect the exact location of interchromosomal translocation to find SVs, and also provide an efficient parallel algorithm using both multi-thread environment, which we used OpenMP in this research, and SSE instructions to find search a large amount of genomic data. So this is the pipeline overview pipeline of the our method. The unaligned BIBM and FASTQ files is the input. At first, we're gonna discover discordant mates and pass these reads to the binary fingerprint generation phase. And we have a parallel Jaccard distance computation phase that calculate the Jaccard distance between pairs. And we are using Hadoop MapReduce to find the nearest neighbors in this research. And the last step is the refinement. And it is the place that we are extracting SVs among the candidates. So the first step is this discordant mate. The input is an unaligned pair and a short read in a SAM format. And we want to align this input to the reference genome and detect the discordant reads that are good candidates for SVs. We are using bow tie in this research. And the distance between the pairs are at least 550 base pairs in this research. The next step is to generate binary fingerprints. So what is a binary fingerprints? Binary fingerprints is an array that contains the read, and we are using this binary fingerprint to reduce the dimensionality of the data and speed up the computation. So you can see in this picture that we divided the read into windows of length 32, and using the Hamming code, binary Hamming code, we can convert these 32 length window into 128 bit binary fingerprints, and we can load this binary fingerprint into an SSE instruction and do the calculation in registers, which is fast. The next step is to calculate the Jaccard distance between input fingerprint and reference fingerprint. You can see that in this picture that we are using population count instruction, which is implemented in using SSE instructions to find out the Jaccard distance. In order to increase the speed of the computation, we are using a tiling mechanism here. We are loading a chunk of data from input fingerprint and also reference fingerprint into the cache, and using several threads, which we are using 24 threads in this research, to calculate the Jaccard distance in a block. So this method is helpful to increase the speed up, because the bottleneck is in memory all the time. And so using loading the data into the cache, we reduce the memory and cache communication, and we can do the computation much faster than a normal serial code. The next step is to find the nearest neighbor using MapReduce. MapReduce used to combine, sort, and find the nearest neighbors of a given query. And the Jaccard distance are proceed, and the most similar samples to a given query have been selected in this phase. The next phase is to refinement phase. The result of the previous phase contains large amount of false positive matches due to sequencing errors. And this phase minimizes the number of the false positives by aligning the resulting reads against the human genome using BLAST sequencing alignment. And we are using local BLAST in this research, which is much faster than a remote BLAST. And this refinement phase considers only reads that are mapped to different chromosomes, which are the definition of the translocation and SVs. So the dataset that we use in this research contains 3 DNA samples of thyroid cancer patients known to have a specific arrangement between chromosome 2 and 3. These datasets are referred as MP6, MP13, and MP14, generated by Illumina high sequence system. And this dataset contains more than 195 million pair in reads. This picture shows the results of our method. You can see for MP6, MP13, and MP14, with decrease of the Jaccard distance threshold, we have more candidates to work with. And the result of it is that we are discovering more SVs. And we observe the same behavior for the number of hits before the refinement phase and after the refinement phase. And this shows that whenever we increase decrease the threshold, the number of SVs would be increased. This picture shows the effectiveness of our parallel implementation compared to serial code. As you can see, on the green line shows the execution time of our parallel method, which is much faster than a serial code. We are compared the performance of our method with the other related method. You can see that on the first row is our method, and you can see for MP6, MP13, and MP14 we discovered SVs, and in all samples our method discovered 82 SVs compared to Delhi, which does not recognize anything. And TIDE is another method use approximation, LSH approximation, discovered only 24, and Lumpy is discovered 162 SVs. You can see Lumpy has discovered more SVs, but it takes much longer time to achieve that. So our method has a good accuracy with a good running time. So to conclude this presentation, we propose a method that identifies location of interchromosomal breakpoints and detect SVs. We introduce binary fingerprints generation that reduce unnecessary redundancy and help us to improve the speed. And also we use a combination of parallel computing, OpenMP, and SIMD instruction to calculate the Jaccard distance and solve the exact nearest neighbor problem. You can see some of references of this research, and I hope you enjoyed my presentation. Thank you so much for your attention.